Welcome everybody online and also in the hall. It's like 50-50. So we have a nice attendance also in the hall, which I didn't really expect. And welcome to the continuation of the seminar series of the INTERMICRO project. We had a break during the summer, and then we had a meeting in Liblice. So we will start now and continue until summer next year. We will try to keep the same time. That means the last Monday each month at 1 p.m. And we hope that we will also be able to broadcast it to everybody from the other partner institutions or those working from home office. I mean, when we were trying to set it up, I mean, it was very late. So I asked Anukool, who actually presented one of his nice papers in Liblice, but he got another one, which was published earlier this year. So I asked him to start up because there was not enough time. But so far we have got a couple of volunteers to continue, but there's still empty slots. So if you feel like presenting during the next year, I mean, any suggestions is most welcome. So I stop talking now and I pass the word to Anukool, who's coming from my group. He's staying with us for more than 1 year now, and we really enjoy developing the finalizing papers together. I mean, that some of them were languishing for a long time, but this is a nice one. So the word is yours, Anukool.
Thank you, Jan. And thank you, all of you, for coming here, as well as joining in online. So today, I should hide float. That's okay, huh? I guess it is visible for online people as well. So today, I'm going to present a work that is published. You can see Applied Soil Ecology, and this is the part of work package 4 in INTERMICRO project. So here we will discuss about the foraging behavior of arbuscular mycorrhiza fungi when they coexist in a single host plant root. So before moving further, I would like to acknowledge all the co-authors in this paper, as well as our lab members who contributed somehow in other way. So I will start with some quick background of arbuscular mycorrhiza fungi. So it's oftenly known as mother of plant root, and it shows a intimate and ancient relationship with most of the plants that are present on the earth. So it's a obligate biotroph that needs carbon from its host plant, and in return, it gives nutrient to the host plant. In fact, it's reported that 80% of the plant nitrogen and phosphorus that is received by the arbuscular mycorrhiza fungi. Now you can understand that how this is important to understand the foraging behavior of mycorrhiza fungi, because when it uptake the nutrient from the soil, then it transfer to the plant, host plant. That's why here we are concerning about the food choice, the foraging behavior of the mycorrhiza fungi. And in the second thing, we also want to see how they coexist in a single plant host root, because in nature, many AMF species can colonize a single host plant root. So we want to see either they complement to each other or compete to each other, and how they foraging on different nutrient sources while they coexist. So with this background, we worked on these 3 different research questions in this study. How do multiple mycorrhiza fungi species coexist when inoculated together? Do they forage for nutrient resources, different nutrient resources, and who are their helpers? Helpers means, like I mentioned, that they are oligotrophs in nature. They don't have extracellular enzymatic activity, that can degrade the organic compounds. So they always need some helpers from soil that we call soil microbes or other microflora. So they always need some helpers for degrading organic compounds and uptaking nutrients. So here we want to see who are their helpers when they start to forage on nutrient sources. And how does this affect plant growth and nitrogen uptake? So for this study, we design a microcosm ecosystem with 10 liters of compartment, 10 liter of plant pot, where we use different compartmental system. You can see here, the middle one is plant compartment. This one is the plant compartment. And beside that, there are 8 different compartment that we call hyphosphere compartment. In the middle compartment, we use andropogon plant. And these different 8 hyphosphere compartment, we use different nutrient patches. That are, some of them are organic nitrogen rich, some of them are organic phosphate rich, and some of them are inorganic nutrient rich. You can see like chitin, DNA, and clover, they are organic nutrient patches. NP control, that is inorganic nutrient patches, and we also place one blank where we didn't use any nutrient patches. So here our idea was that once we inoculate our mycorrhiza fungi in the plant compartment, where we use 5 different fungus, Rhizophagus, Anthropospora, Funaliformis, Zygospora, or Echilospora. So when we inoculate these fungi in the plant compartment, they will start, we suppose that they will start to migrate outside the plant compartment. And we cover this plant compartment with 40 micrometer nylon mesh. So we can create a barrier to pass the root outside this compartment. So we can create hyphosphere compartment, hyphosphere environment outside this main plant compartment. And there was one more treatment without mycorrhiza. So we can compare the effect of mycorrhiza without non-mycorrhiza. And we check their abundance with 2 different methodology. One is qPCR, quantitative real-time PCR, and second is amplicon sequencing through Wanda AML2 primer. There is a difference, like qPCR primer, they are dependent on the larger ribosomal subunit, and the Wanda AML2 primer, they are dependent on the smaller ribosomal subunit. So we use both of the approaches for quantifying the amplification of mycorrhiza in different compartment zone. And we also trace the 15N transfer, the nitrogen transfer, by using isotopic labeling 15N. And for those labeling, we use chitin and clover as a nutrient patch. So we labeled our chitin and clover with 15N isotope. And we trace that 15N isotope, we trace the transfer of 15N from hyphosphere to the plant. And we also check hyphosphere microbiome profiling, as I mentioned earlier, that we want to check who are their helpers during foraging time. So we also check the hyphosphere microbiome profiling. So what we found in our result, we clearly see a difference between mycorrhiza and non-mycorrhiza plant. Mycorrhiza inoculation significantly enhance the plant growth as well as the nutrient content. You can see, like in the right hand side of, like here, you can see the transfer of 15N from clover and chitin in the mycorrhiza treatment. That shows that yes, mycorrhiza can uptake nitrogen from beyond the root and can transfer to the root, because these clover and chitin, these nutrient patches were present in the root-free zone in the hyphosphere compartment. So it clearly seen that mycorrhiza help for transferring nitrogen from hyphosphere to plant compartment, as well as they also increase the total phosphorus and total nitrogen content, and also they also increase plant biomass content. Now come to the main finding, the AMF colonization, that we want to see how they coexist. So if you see in the root region, we can clearly see the competition between 5 different AMF fungus. We clearly see the competition, the higher abundance, the higher colonization was performed by the Rhizophagus in this red color bar, and then the Funaliformis. And the lowest was done by the Echilospora. So we can clearly see the competition in root colonization. But when we come to the rhizosphere, we found some equal level of all 5 AMF fungal colonization. And when we talk about the root free, only two fungus, Phenoliformis and Rhizophagus, can come out from the root compartment, from the plant compartment to the root free compartment. So only two fungus, Phenoliformis and Rhizophagus, out of the 5, they come for the foraging outside the plant compartment. And here we also see a difference in our both approach, like qPCR analysis is showing absolute abundance, and Vanda ML2 sequencing, they are showing relative abundance. So you can see, like here in the rhizosphere, in Vanda ML2 sequencing, we found higher colonization with Enterophosphora, but in qPCR, we found higher colonization with Rhizophagus. So there was a difference between both of the approach, and that is because the larger subunit, if you use primer based on those target larger subunit of ribosome, because they have higher variability. So they can be choose for close species, to identify close species. But because Vanda ML2 is dependent on the smaller ribosomal subunit, so that's why it covers most of the species that we use here. So that's how we found differences between both approaches. But you can see the clear, like here again, the only two fungi that comes out from the plant compartment to the root-free compartment. And when we check the foraging behavior on nutrient patches, we found that the both fungi, Phenoliformis and Rhizophagus, they clearly show a resource partitioning in foraging on different nutrient patches. Like Phenoliformis choose chitin and DNA, and Rhizophagus choose clover for their foraging. We found higher abundance of Phenoliformis on chitin and DNA patches, while Rhizophagus was higher on clover patches. That shows that they have different preference for their food when they start to foraging. They have different preference for their food. And here, one more thing you can notice, that Rhizophagus was also present on poor nutrient patches, like in cellulose, that is only carbon. Like in blank also, there was nothing, no nutrient patches, only soil substrate, as well as in starch. So Rhizophagus was tried to explore on all nutrient patches, right? And Phenoliformis choose organic nitrogen-rich patches for their proliferation, that we found in when we check the relative allocation of their biomass. Here you can see that both genera prefer organic nitrogen-rich patches for their foraging, and in Phenoliformis, it covers about 75% of their total biomass in organic nutrient patches. And in Rhizophagus, they almost cover 65% of their biomass, and rest on lower, or you can say the poor nutrient patches in Rhizophagus case. So that represent that both fungi have their different preference in their food choice, and that how they perform resource partitioning, so they complement to each other. They survive there, they do not compete, and that's how they provide maximum benefit to their host plant. We also check 15N partitioning. That's where we try to find the loss of nitrogen from soil. So in about 2 pie chart, you can see, like when there was no mycorrhiza, the 48% loss from chitin. The loss of nitrogen was 48%, and the loss of nitrogen was 23% from clover compartment, when there was no mycorrhiza. But once we inoculate our mycorrhiza in the pot setup, the reduction, the loss of nitrogen was completely reduced, not completely, but it was reduced up to 26%. And here in the clover compartment, it was reduced up to 7%. And the rest of the portion was transferred to the plant, like this pink one. The 39% nitrogen was transferred to the plant, but it was not here because there was no mycorrhiza. And again, same here. So this analysis also suggests that when we inoculate our mycorrhiza, or where the mycorrhiza in the soil, they try to retain nitrogen in the soil as much as possible. That's one of the method to discuss that how we can retain nitrogen, or how we can reduce the loss of nitrogen once we introduce the chemical fertilizer in the soil. So this finding also also suggests that AMF inoculation can reduce overall nitrogen loss. And now come to about the helpers, microbial helpers that help to mycorrhiza for uptaking organic nutrients. So here we choose hyphae, like these are the runner hyphae who comes out from the plant compartment outside the root-free compartment, and they try to forage on these nutrient patches. These are two different nutrient patches. So we choose these runner hyphae as well as hyphae, those are present in these compartment, nutrient patch compartment. And we try to see which type of microbial community they have on their hyphae. So we found that the mycorrhiza hyphae change bacterial and protist community when they start to forage on nutrient patches. They change the bacterial and protist community. So there was some enrichment in bacterial community. There was some member like Pseudoarthrobacter and Nocordioids, and in protist, Paraparentocoirus and Straminophiles, their abundance was enriched in the presence of mycorrhiza when they try to forage on hyposphere compartment, nutrient patch compartment. So what it reflects that somehow these microbes, these bacteria and protists, somehow they help in nutrient mineralization from organic sources, and then mycorrhiza uptake those nutrients and transfer to plants. That's what I present in Liblice conference, where we try to find how protists degrade protein and release the nitrogen from protein that further uptake by mycorrhiza. So here we found that yes, when they start to forage outside the plant compartment, they carry their own microbe for helping in nutrient uptake. So they are also present in runner hyphae here, and they are also present in here compartment. So we can assume that, yeah, maybe they are required for the nutrient uptake. So our key findings are like AMF diversity. We use 5 different AMF fungus. So their diversity enhance plant nutrition in complementary way. So here I use this picture because like functional complementarity, we can understand by a musical band where every musician has different instrument and they play together to harmonize the music. And that's how every mycorrhiza community work together to provide maximum benefits for their host plant. This is functional complementarity. So that's functional complementarity we found in our AMF diversity, and that's how they give maximum benefit to the host plant. In the second finding, Rhizophagus and Phenuliformis were dominated in root colonization, as well as they can forage beyond the roots. And during foraging, we found resource partitioning. That's clearly seen that one fungi choose different compartment and one fungi choose different compartment for nutrient foraging, for their food choice. And AMF enriched some microbial helpers like protists and bacteria during their foraging. And also AMF inoculation reduced overall nitrogen losses that we found in from 15N partitioning from clover and chitin compartment. When we inoculate the mycorrhiza, the losses of nitrogen was reduced.
Also, maybe. You better stop.
And finally here,
What else? Yes.
so here,
There will be a blood feud between us. What? Blood feud. It means I'm going to come for your family too.
Please mute your
That's
microphone.
the nitrogen line. What?
Keep going, guys. It's great.
Yeah, that's fine. So here we conclude our all findings where we present some explanation why we found this type of observation. So we start from here, like in root colonization, we clearly see competition. So we suggest two explanations for those competition, like it might be because of the host filtering. Host filtering means root favor only those fungi that can provide maximum benefit to the root. So like Funiliformis and Rhizophagus, they go beyond the root and uptake nutrient and transfer to the plant. That's why their abundance was higher in root as well. So that might be one explanation. And second explanation might be interspecies competition, because we choose different family species and there might be a natural interspecies competition. That's why we found like Glomeraceae family, like Funiliformis and Rhizophagus, only those were higher in root colonization and other were lower in the colonization abundance. So these two explanations may be possible for showing the competition in root colonization. In rhizosphere colonization, we found like all AMF fungi, all 5 AMF fungi were similar in higher abundance. So we can say that it is because of the insurance effect. The insurance effect means the higher biodiversity ensure the stability of the functioning, the stability of the ecosystem if something goes wrong in the ecosystem. Or maybe we can understand this by a sports team, they always prefer to use extra players, substitute players. Like, so if anything goes wrong during the match, the substitute player can step in and try to maintain the performance of all the team. So likewise here, the higher biodiversity ensure that if any fluctuation, any disturbance occur in ecosystem, they can maintain the ecosystem functioning. So that's how we found the common, or you can see the equal balance, equal abundance of all fungi in the rhizosphere. And when we talk about the foraging on root-free soil, there might be two explanations. One is mycelial architecture. Like Zygospora has thicker hyphae, and we use 40 micrometer mesh to create a barrier around our plant compartment. So it might be possible that because of their size of hyphae, they cannot pass out, they cannot migrate from plant compartment to the root-free compartment. And the other explanation might be niche partitioning, that some fungi like stay nearby the root, like they are dependent on the root exudate for their nutrient uptake, and some fungi like to forage on different nutrient patches. So there might be two possible explanations when mycorrhiza come out from the plant compartment to the root-free compartment. And in foraging on nutrient patches, we clearly found resource partitioning, where Funiliformis choose DNA and chitin for their nutrient uptake, and Rhizophagus choose clover for the nutrient uptake. That's all from my side. Thank you. Thank you for your attention.